Je vais donc introduire le premier orateur que le professeur de Mulder a déjà annoncé également. Il s'agit du professeur William Morice. C'est un grand plaisir, un grand honneur de le voir participer à notre réunion. Donc le professeur Morice est hématologue, biologiste et professeur de biologie clinique à la prestigieuse Mayo Clinique. C'est un chercheur reconnu dans le domaine du diagnostic des maladies du sang, des leucémies en particulier, et plus précisément les troubles lymphoprolifératifs des cellules T et des cellules NK. Mais ce n'est pas de cela dont il va nous parler. Il va plutôt nous présenter sa vision et la place actuelle et à venir de la technologie, telle que le big data évidemment, dans le contexte de cette pandémie qui, c'est amusant si on veut, l'a empêché d'être présent avec nous, et donc sa présentation est enregistrée, et étant donné le décalage horaire, malheureusement le professeur Morice ne pourra pas répondre à vos questions. Voilà, merci.
Hi, I'm Dr. Bill Morice, the president of Mayo Clinic Laboratories and the chair of laboratory medicine and pathology at Mayo Clinic in Rochester, Minnesota. It's really an honor for me to be invited to present to you today at the symposium. I really regret that I can't be there in person because I'd love to visit you and your country and be there, but unfortunately, given the pandemic, I can't at this time, but I look forward to doing so in the near future. What I thought I could talk about today is really thinking about the pandemic in stepping back and just how we've approached it from a diagnostics perspective, particularly in the United States. So to give you some background, some context, really I'd like to explain just a little bit about what Mayo Clinic Laboratories is. So Mayo Clinic Laboratories was actually founded in the early 1970s. And what it does, it allows us to take the tests that are available in our academic pathology department and really at that point just open them to hospitals in our part of the country of the United States. What it's grown into over the subsequent 50 years is actually a global reference laboratory that serves over 4,000 hospitals in the US and actually is a global company as well with activities in over 80 countries. And it has grown to the point that it is now the third largest reference laboratory in the United States. And it's part of that, a large part of that is because the tests that we offer are the same ones that we offer for our patients at Mayo Clinic. So they span everything from pediatrics to adult disease and cancer and hereditary disease, everything. So we have a very complete test menu, and we make those available to anyone who uses Mayo Clinic Laboratories. And it has grown to the point where in 2019, pre-pandemic, we did over 25 million tests on our campus. And we actually have the logistics, since we do all the testing in Rochester, to insource over 50,000 specimens a day, as we learned during COVID. Most importantly, perhaps, is that through our Mayo Clinic Laboratories and my role in Mayo Clinic Laboratories, I actually was in Washington, D.C. in March, in the first week of March of 2020, as part of the American Clinical Laboratory Association, which is the trade association for reference labs in the U.S., and I was actually at the White House visiting with Vice President Pence and the COVID response team in those early days. And so really Mayo Clinic Labs has been kind of right at the epicenter for testing for the United States for COVID. And it has not just for our own patients, of course, but for those across the country. And so it's given us sort of a unique perspective on this, which I'd like to share with you today. And I think it's really kind of informative when you think about how we've managed testing through the pandemic. As you go back and look at the last really global pandemic of this significance, which would be the Spanish flu of 1918. And if you look back to that pandemic, there's actually a lot of similarities, things that ring very familiar to us who have lived through the time of COVID-19. Of course, the Spanish flu was caused by influenza, the H1N1 virus. Its origins were unclear. It actually probably started somewhere in the United States, probably in Kansas. And as we know, it is now an endemic because the H1N1 strain of influenza still circulates and causes illness today. This first appeared in the spring of 1918, had a second wave in the fall of 1918, which was much more lethal. The big difference, thankfully, from COVID-19 as compared to the Spanish flu or the H1N1 pandemic is that pandemic seemed to really have greater lethality in younger people. particularly in their late teens and early 20s. And so that's much different, obviously, than COVID. It had a high mortality rate, somewhere between 10 and 20%. So 10 to 20-fold higher than typical influenza. Again, not too dissimilar in some ways from COVID, although COVID thankfully doesn't have that high of a mortality rate compared to other coronaviruses. It is estimated to cause somewhere between 20 and 50 million deaths globally. Interestingly, in the United States, estimated to have caused over 600,000, and that's pretty close to the number now that we are at for COVID-19 in this country, although, of course, in the United States, the population was much lower, so proportionally it was higher. It ended in 1919, and no one really knows why, but it apparently is due to widespread acquired immunity, and there were no effective interventions which were ever identified for that disease at that time. And now compare that to the timeline here for the early timeline, at least for COVID. We first started hearing about pneumonia of unknown etiology in Wuhan province, Hubei province, Wuhan city of Hubei province, China, in December of 2019. Early January of 2020, the novel coronavirus, now known as SARS-CoV-2, was identified by the Chinese CDC. First recorded death was in early January in China as well. And as you can see from there, it essentially rapidly spread across the globe. And where by April of 2020, we had over 3 million confirmed cases. And really in January 30th, at the end of that month is when we looked at a global health emergency being declared. United States, it was that first week of March, early March, that we just declared a health emergency in the US. So this spread very rapidly. And you can see there in March is when it was actually declared a pandemic. I don't know if any of us at the time anticipated that it would be so, what kind of timeline we've been on as we're now into late 2021 and still trying to work our way through this global health crisis. And it is a global health crisis, and to really think about, how testing has been central in our response to that is really you have to step back and think about this, the pathogenesis of the virus itself. I'm sure this has been discussed and familiar to most of, not all of you, so I won't go into any great detail, but like any viral infection, it has a period of viral replication, followed by inflammation where there's systemic illness. We now know that the spike protein binds to ACE2 receptor, and that the pattern of inflammation and the pattern of symptoms and signs and sequelae of viral infection very much follow patterns of ACE2 receptor expression in terms of cardiovascular effects as well as respiratory and GI effects. We also know a lot more that compared to starting from essentially zero information in January of 2020 to where we are today. We know a lot more about the viral replication, now about viral evolution, and also about the immune response, although amazingly enough, there's still a lot to learn in all those areas, right? And this is really a kind of a schematic of what is thought to be the typical response to SARS-CoV-2. Again, very similar to what we would see with other viral infections where there's an early period of viral replication. Then after about a week, you can start to see an immune response with the serologic conversion. Seroconversion first with IgM, then with IgG. Typically it correlates with when symptoms appear, and then the patient enters into, I should say, a convalescent period. So to step back and think back to the conversations, even though those early days of March at the White House, and kind of how COVID testing was going to be used to help both the United States and the world manage the pandemic, there's really three applications when you think about for viral testing. The first and the most, obviously the most pressing in those early days and still today is a diagnosis of symptomatic patients. Now, the challenge with that was that we had this disease that was rapidly spreading, and yet the tests that had only newly been created. And so the demand for testing far outstripped the supply of testing. And that was something that we really struggled with really throughout most of 2020. There's the other applications though for testing that we have to bear in mind. And again, they were prescient even at the onset of the pandemic. One is the screening for infection. So identifying those who are infected and maybe don't know it. And this is a real challenge, as we'll talk about, because we now know that those who are most apt to spread COVID actually are those who are pre-symptomatic. And there was a lot of debate about asymptomatic spread and controversies around that, but there's no doubt that pre-symptomatic spread is an important part of the disease propagation. And last but not least was a lot of dialogue in the early days about surveillance for outbreaks and surveillance and suppression of outbreaks. That has been successful in some parts of the globe, particularly in Asia, where they had systems in place to do that. We can talk about that in the United States in particular, which is my experience, of course. We have not been particularly successful with that, and we can talk a little bit about why that might be. So here's, again, this is pre-Delta even, so older data, but I think is a very informative diagram showing the days after exposure and then the probability of a positive test. And so day 0, of course, the probability after exposure, the probability of a test being positive is almost 0, if you will. And then by the time a patient develops symptoms here, as shown with the dotted line, they were about 70 to 75% likely to have a positive test by the methodology that we were using at the time, which is still very good, and that's the centralized lab testing. So there's a period of time of about 5 days after exposure where the patient, the individual, has a virus, and yet they don't know they have the virus. And unfortunately, it is in this really immediately pre-symptomatic period that individuals seem to be particularly apt at spreading the virus. And this is really much different than SARS-CoV-1 or MERS, which in patients who were symptomatic appeared to be those who were most apt to spread it. And that probably is a major reason why we were able to suppress global outbreak of those coronavirus illnesses, and we have not been able to for SARS-CoV-2. The other thing, though, that is really interesting about that pre-symptomatic transmission is the stress that it put on the diagnostic infrastructure in this country. And that is, we already knew we didn't really have enough tests even for symptomatic patients to confirm they had SARS-CoV-2. But now, from a screening perspective, to identify those who might spread it, you're now needing to actually test everybody because you can't rely on symptoms to drive when you might screen someone for possible infection. What that led to, of course, then was a lot of debate about how much virus, what was the viral load of someone that was pre-symptomatic that would make them apt to spread it. There were many controversies and even recommendations about how to use a CT value from a molecular PCR-based SARS-CoV-2 test to help, what was the right threshold for calling someone positive, if you will. And this is from our own laboratory, just basically showing that our cutoffs, like most labs, I would think, were predicated on, basically the analytic validation of the assay and using limiting dilution to know how far we could go compared to a control, which is shown in yellow. And we got down to very low copy numbers, but where you could see we had significant difference between the control and the target, even at a CT of greater than 30 or 35. And then there was debates about it. Was that too high? Of course, this is not, at best, semi-quantitative analysis. And so really, it shows the dangers of trying to take something that's not designed for the purpose that people are putting it to. And that is taking a semi-quantitative, at best, PCR assay for SARS-CoV-2 and trying to make quantitative assessments. And so what we did in our own laboratory was actually try to correlate in individual specimens the CT value to the actual viral load as determined by digital droplet PCR. And that is shown here. And it's really just for beyond crossing a CT value of 29. And that's just to show that true enough, at very high CT values, if you it's shown on the x-axis, on the y-axis, you can see the viral load was very low. However, if you work back from that, even at 31 cycles, but especially at 30 cycles, you can see we have individual specimens which have very high viral load. And it really shows the danger of arbitrarily picking a CT value to actually understand how much virus, what the viral load might be of the individual. Again, really earmarking some of the limitations of our current strategies around detection and how they may be used in terms of making decisions and screening. So, of course, now, particularly in the United States, there's a big focus on point-of-care testing, particularly rapid antigen testing, as a way to screen individuals for potential infection. This is being invested in by the federal government. There are protocols being written to what should be done by employers, particularly if their employees are not vaccinated, how frequently they need to test. The reality is that rapid antigen tests have not been the mainstay of detecting respiratory viral infections for a number of reasons, not the least of which is their challenges around sensitivity. There's also a push now for point-of-care nucleic acid amplification tests, like LAMP-based technologies from companies like Q. These also have limitations. The real limitation on the rapid antigen tests is around their sensitivity and their performance. And to think about the performance of these tests, it's also really important to think about the prevalence of the disease. And that's something that outside of laboratory and public health officials, I think, and even amongst public health officials at times, can be a difficult concept to grapple with. And that is, if the disease prevalence is low, even if the specificity is relatively high, as is shown in this chart, if you will, that more than likely you're going to have a false positive test, even from something that has a relatively high specificity, like a 90% specific antigen test. Conversely, if you look at if the disease prevalence is high, sure enough, you're going to have fewer false positives, but then again, you're going to have more false negatives. And so what really needs to happen is we need to think about, from a precision perspective, how to use the different modalities together to accomplish the goal of screening for people who might be at risk of spreading SARS-CoV-2, even in a world where we don't have complete vaccination or complete immunity. And the reality is that we're going to have a combination of tests, probably antigen as well as in-lab PCR, to really do the best job. And yet we still don't have these systems in place. There's been a lot of discussion, data from academic groups like Imperial College in London showing and modeling how these tests need to be used, but we still don't have the system in place to actually give individuals the best answer. And it is a concern for us in the US as these rapid antigen tests are used more and more, how we're going to actually make sure that the information that people are getting is accurate and actionable, and how we're going to use labs like a Mayo Clinic labs and an academic in-lab test to confirm some of those studies. So in terms of looking back now over 21 months of testing for SARS-CoV-2 for Mayo Clinic, and then our participation in the national response, two things: scalability is a real issue. And as we look ahead now to pandemic preparedness in the future, thinking about both supply chain as well as the regulatory process by which tests are reviewed and approved, both in the US and globally, becomes really important. And I think one of the things that we have seen and experienced, all of us, is the fact that each country has essentially had to find its own way to respond to a pandemic which was spreading much more quickly than we could adapt to. Is something we're going to have to consider as we look ahead to how we're going to do things better in the future. Number two is that if you look globally and within the US, clearly the availability of testing is a driver of healthcare outcomes. And so to start to think about equitability of access becomes very important. Again, it was part of the global summit, which I had the opportunity to participate in, organized by President Biden here in coordination with the UN Global Summit earlier in the fall. It's something we're going to have to continue to look at. And last but not least, we still don't have the systems in place to understand how we use all these different tools: rapid antigen tests, point-of-care molecular tests, in-lab tests, to best manage for individuals and for groups of individuals or societies. How do you manage this pandemic? Still a lot of work to be done in terms of how to use our diagnostic tests to their greatest ability or their greatest precision, if you will. In terms of precision and action, the other thing, of course, is surveillance. There has been a lot of talk in the United States about the best way to do this. Wastewater epidemiology is a known tool in surveillance for viral infections. And it was something that has been of interest, and that we have collaborated with investigators at the University of Minnesota to look at for its utility in our own state. Minnesota is an interesting state to look at because the population density is quite variable, as is shown in the map of the state, where red, you can see most of the individuals live in the Minneapolis-St. Paul area, whereas the remainder of the state, the population density is quite low. And I think that mirrors many other parts of the world, including Europe. And so the question is then, how would you normalize when there's all this different population density? How could you actually normalize the data that you might get from wastewaters to know it's not just a correlate of the number of people who are served by a wastewater facility? What we've worked on, and others have talked about this too, is there has to be some kind of control virus that you look to. It turns out pepper mild mottled virus is something which is found actually across the U.S. I think it might be quite ubiquitous across the globe, but I don't know that for sure. But that serves then as a normalization control to have something to baseline a SARS-CoV-2 signal against in wastewater. And that's really what we've studied then, and its ability to do this. And the bottom line is that if you normalize to something like the pepper virus, you can get about a 2-week insight into where you're going to see a rising number of cases. And that's shown here. So the actual case numbers in Minnesota are shown in the gray, and the solid line is the wastewater. And you can see it mirrors very well the rise in cases with about a 2-week anticipatory window. So we can do it. We can do this in other ways as well. Mayo Clinic developed its own predictive models that we used both within the state and in Arizona and Florida, where we have campuses and across the country, and looking at things even like Google searches. There's a lot of metadata out there that actually correlates quite well with where we're seeing outbreaks or rising incidence of cases. So there's lots of ways to do it, but there's no standard method. So each state and each part, because in the United States, each state has its own Department of Health with public health concerns. And so there was no standard across the United States. Yet, even probably more problematic, there was no system in place to actually respond to that information. So in Asia, in countries like South Korea and Singapore, where they have systems in place to actually intervene, China as well, when they could see this information, they could use it to great effect. In the United States, we do not. And I think it's something we're going to really look at as part of our pandemic preparedness, both in the United States and globally in collaboration with the WHO. Another huge ongoing question is immunity to SARS-CoV-2. And again, this is the schematic showing the injection in the purple, in the yellow, in the green and yellow, green and red, antibody production, and then the yellow, the symptoms. Now, or IgA, So essentially, this is sort of the typical antibody response you would see to a respiratory virus with early rise in IgA and IgM and conversion to IgG. One thing we learned early on with the pandemic is that this paradigm was not routinely followed by individuals, if you will, after they were infected. There certainly was a correlation between the degree of symptoms and the strength of the antibody response. There were also with patients that appeared to never develop an IgM response at a minimum. IgA is very difficult to measure, and so was not routinely measured. And so what we saw essentially was a lot of variability in the serologic response to SARS-CoV-2 infection. That being said, there was a lot of talk about how we might use serologic information to help understand where a patient was in terms of being infected with SARS-CoV-2. That's shown in this graph here, or this chart here, where we had different sorts of algorithms you could use to determine if someone might be infected. There was a lot of talk about this, particularly when the PCR tests shown there in the left column were in short supply. Again, there's no even unanimity in the infectious disease community about how applicable or consistent something like this could be used. And so really, I don't know, at Mayo Clinic at least, and I don't think in many medical centers, this was not routinely employed in terms of understanding and diagnosing SARS-CoV-2 infection, even though the diagnostic tests were in short supply. Serologic tests were available in the United States in early April, including at Mayo Clinic. We anticipated a significant surge in demand, similar to what we saw with PCR, and it did not materialize. And it's probably still been about 1/10 of the volumes that we've seen compared to molecular diagnostics. And I think there's a lot of reasons for that. Some of them are universal. Some of them are more specific to the U.S. One is that providers, doctors don't really know what to do with the information, kind of back to the last slide. So they're much less apt to order it. They don't really know there's still no great studies almost 2 years in to say what degree of seropositivity correlates with protection from infection. We still don't have that kind of basic information that can guide providers and patients in terms of, are they immune to COVID? Number two is that in the U.S., unlike the tight regulation around PCR, which limited its availability, a more open approach was taken by our FDA in making these tests available. What happened was we had poor quality tests that made it onto the market, which actually undermined the public confidence, not just in serologic testing, but all testing. So when I was participating in press conferences like with the governor of Minnesota, and when we were working with him and the University of Minnesota to make COVID testing available to the state, there were questions asked about the quality of testing, just because in the public mind, testing is testing is testing. So there was an undermining confidence, particularly in these tests. There was a lot of thought that we'd use serologic testing to understand immune response after vaccination. Two things really worked against that. Number one, the speed with which we were trying to get EUA approval of the vaccines. So there wasn't really time to design studies with a lot of serologic follow through. The other was the high efficacy of the vaccines in the early studies, over 90%, meant that there wasn't a lot of trying to understand those who were not responding. Although clearly that is a concern now, as we know there are patients that don't respond or have a suboptimal response to the vaccines, particularly those being treated for cancer and other things. Bottom line is we still don't have a clear role of diagnostic testing to understand who's immune to COVID. There's the T-cell response test available in the United States from Adaptive Biosciences, which gives a different level of information. Even there, I don't think the utilization is very high because they have good correlative studies, but still we don't know what it means for individuals. So this is something that we'll really have to grapple with. We think about the age of precision medicine and coming into before the pandemic, focusing predominantly on cancer and how you could really understand a lot about someone's malignancy and their response to it. And yet in this pandemic, we still don't have a precise way to measure immunity to the virus, which is a fundamental concern really will be a fundamental concern going forward, even when we get into the endemic phase of this virus, hopefully sooner rather than later. The one area where serologic testing was used and studied within our institution was in the concept of convalescent plasma, which of course goes all the way back to diphtheria as an effective way to battle infectious disease, and that is taking serum from a recovered individual and using it as a therapeutic. In recognition of this, it was really Dr. Mike Joyner at Mayo Clinic, along with Dr. Arturo Caspidel at Johns Hopkins, which had the idea in late March to have a national expanded access to convalescent plasma program that could be instituted. Mayo Clinic played a central role in working with the FDA and with Johns Hopkins working with the FDA for Mayo to have a centralized national study with institutional board, review board approval and a central data collection at Mayo. And by April 3rd, this was approved and was rolled out. And within a week, actually, we had the first patient transfused. This is a map. It was actually originally designed we were going to have about 5,000 patients that we wanted to evaluate across the country total to try and understand the potential usefulness of convalescent plasma in fighting COVID. As you can see from the map of the U.S., this is a bubble diagram here on the left showing it was a national program. You can see a lot of activity in the Northeast and the New York area around this, but also a lot in California and Texas and Florida, all across the country. The bigger the bubble, the more patients that were registered and transfused. And you can see we had a lot. And on the right, it shows the effort that was made in terms of collecting the plasma, delivering the plasma to hospitals across the country. And what was originally designed for 5,000 patients actually ended up with over 100,000 patients who were transfused. Now, the challenge with that was that we had a lot of patients transfused, but the data collection was difficult. Especially what we learned is it was very much feast or famine, i.e. There were hospitals that had very few COVID cases, or there were hospitals that were so overwhelmed with COVID cases that even if they had plasma and they transfused plasma, they had a hard time actually recording the data because it was all manual. And also, this was really it was not an efficacy study. It was really to make it available and to just determine if it could be used safely, which clearly it could. That the data showed that the convalescent plasma was safe. It was more challenging to just show that it actually had efficacy because the trial was not designed to prove that as an endpoint. But what we saw was that there were correlation between convalescent plasma use and preventing mortality. That's also shown here. Basically, higher antibodies concentrations and also given earlier in the disease course. Both of those correlated with a decrease in mortality. This led to the emergency authorization of convalescent plasma in August of 2020. But overall, we never really saw a lot of uptake with convalescent plasma either. There's probably a number of reasons for that, in part because of the challenges in just standing up the program, and those are shown here, and in part, and also delivering the product consistently. Also, again, not any kind of national protocol for how to actually walk through this. And also the emergence of monoclonal antibody therapies. I would say, though, that one thing we have seen as we continue to study convalescent plasma is that it does appear that it continues to have a role, because as we know, those monoclonal antibody therapies available, many of them were subject to decreased efficacy with the emergence of variants like Delta. And we found that convalescent plasma actually overcomes some of those because it's a more dynamic response. So there still is potentially a role, but a lot of things that would need to be planned for in advance if we were to actually have to use this tool again in the future. So the conceptual model was confirmed, but more work to be done to actually make a usable system in the future, again, around how we would actually drive patient-specific therapies in response to an emerging infectious pathogen. Last thing is just to touch on quickly was a lot of energy, particularly in the winter, starting in the winter, I think, of 2020 and into 2021, about looking at SARS-CoV-2 variation or mutation, right? And this really was probably the interest really started with the first variant of concern, now considered the Alpha variant, which was identified in the UK, as well as and that was called P1. There was a variant emerged in Manaus, Brazil, also a variant of concern. At that point, there was more interest in looking at viral mutation. A lot of work had been done up before that showed us that the mutation rate was relatively low compared to influenza, which is typical for a coronavirus. But it started to be important, and where clinicians were wondering if this is something we should start to actually have available clinically, so for understanding patients. Why the variants became concerning were their increased transmissibility, their increased virulence, their ability for immune escape, their resistance to therapy, including remdesivir, and also, if you recall, the impact on sensitivity if the mutations were in the primary regions used for PCR tests. These were all concerns, all been borne out to some degree. Probably the increased transmissibility and immune escape are the greatest concerns, as we've seen with Delta. Delta probably doesn't have increased virulence, but it's increased transmissibility with higher viral load and ability for immune escape are why it's caused such problems across the globe. Similar to serologic testing, however, what we haven't seen is a huge rise in demand for this testing clinically. And that is, even with the emergence of Delta, there was some interest in breakthrough, and there still is. But the reality is that now in the United States, over 95% of the cases are of the Delta variant. So as it turns out, the strains that really have a competitive advantage appear to become the predominant strain so quickly that the need for mutational analysis as a clinical tool is probably pretty low. The need to set up a global surveillance system is quite high, and we're still trying to understand how to do that. I know Africa CDC is working diligently on this. A number of areas of the world are, because clearly what COVID has shown us is that we are a global community when it comes to these variants. And look at the spread of Delta across the globe, even at a time when international travel was much different than it was, in the late winter or early 2020 period when we first saw the spread globally of SARS-CoV-2. So in terms of the lessons that I think we've learned, at least by myself personally, around this pandemic and around SARS-CoV-2, is that there are many effective diagnostic tools which exist, but there's no silver bullet test for transmissibility. And the best way to get precision, particularly in the setting of an emerging infectious pathogen, is probably to think of all the different modalities that we have, both point of care and antigen as well as molecular based, to understand the best approach to try and both identify and diagnose patients and prevent the spread of those who are infected from spreading it to others, or understanding where there's outbreaks. Number two is that we really need to understand the utility of serologic testing and other tests that we have available in terms of understanding who's immune to these viruses. And I think, honestly, we'll still have this need for SARS-CoV-2. Who needs boosters? This is a very controversial topic, both in the United States and globally, where there's much of the world where over 90% of the population is still unvaccinated. So the question about equitable distribution and what's the real need for a booster versus the global benefit of getting a vaccine spread, implemented or delivered across the country. Still, a lot of that could be much more informed if we had better reads on who's actually immune to the virus versus not, and therefore who needs supplementation or prioritization for vaccination. Still, I need to understand the role of pathogen surveillance and monitoring. as a global capacity and global capability. I think we've dodged a bullet a few times. I've been in my role since 2015. Before SARS-CoV-2 in the United States, we had Zika virus emerging out of Brazil. We had Ebola emerging out of Central and Western Africa, and we had a lot of concerns around those and how we're going to be prepared. We dodged a bullet because they weren't nearly as transmissible as it turned out as SARS-CoV-2. But SARS-CoV-2 has taught us that we need some kind of surveillance and monitoring for these pathogens as a global capability. And also, I think I've heard estimates that we can anticipate the next pandemic within the next 5 to 7 years. And the need for some kind of warm infrastructure. I mean, the challenge is when I look back to just understanding we couldn't get swabs in the United States because most of the swabs that we used at the time were produced in northern Italy, which was being in the ravages of its own, being one of the epicenters early in Europe of COVID. So just the understanding of how we have enough global production capacity when we need a test rapidly deployed as a global tool is something we're going to have to look at, not to mention therapeutics. And PPE, personal protective equipment, still oxygen, struggles with oxygen delivery for patients, something we just have to really look at. And then last but not least, of course, is looking at regulatory frameworks, not even on a state-by-state or country-by-country basis, but how would we have more rapidly approved diagnostics that could be used and deployed across the globe with a consistent regulatory system that ensures that they're good tests, right? And that the therapies are effective and safe, something we're going to have to think about. And last but not least, because the time to design a system to respond to a crisis is clearly not during the crisis, but it's before the crisis emerges. Even if you don't need it for years, that investment pays off huge dividends when it actually is needed. If you look at the human costs and the economic costs and the trillions of dollars that have been really unleashed upon us in terms of, as a global community, in terms of human suffering and economic loss. So with that, I hopefully Professor Vandenberg will, if there's questions, that they'll pass them along. Maybe we'll save them for the near future when I'm there. But I thank you for your attention and the opportunity to participate. And I wish you a very successful conference and look forward to joining you in the future.